The cytoskeleton. If you were to remove all the organelles from a cell, would the plasma membrane and the cytoplasm be the only components left? No. Within the cytoplasm, there would still be ions and organic molecules, plus a network of protein fibers that help maintain the shape of the cell, secure some organelles in specific positions, allow cytoplasm and vesicles to move within the cell, and enable cells within multicellular organisms to move. Collectively, this network of protein fibers is known as the cytoskeleton. There are three types of fibers within the cytoskeleton: microfilaments, intermediate filaments, and microtubules. Figure 1. Here, we will examine each. Microfilaments. Of the three types of protein fibers in the cytoskeleton, microfilaments are the narrowest. They function in cellular movement, have a diameter of about 7 nanometers, and are made of two intertwined strands of a globular protein called actin. Figure 2. For this reason, microfilaments are also known as actin filaments. Actin is powered by ATP to assemble its filamentous form, which serves as a track for the movement of a motor protein called myosin. This enables actin to engage in cellular events requiring motion, such as cell division in animal cells and cytoplasmic streaming, which is the circular movement of the cell cytoplasm in plant cells. Actin and myosin are plentiful in muscle cells. When your actin and myosin filaments slide past each other, your muscles contract. Microfilaments also provide some rigidity and shape to the cell. They can depolymerize, disassemble, and reform quickly, thus enabling a cell to change its shape and move. White blood cells, your body's infection-fighting cells, make good use of this ability. They can move to the site of an infection and phagocytize the pathogen. Intermediate filaments. Intermediate filaments are made of several strands of fibrous proteins that are wound together. Figure 3. These elements of the cytoskeleton get their name from the fact that their diameter, 8 to 10 nanometers, is between those of microfilaments and microtubules. Intermediate filaments have no role in cell movement. Their function is purely structural. They bear tension, thus maintaining the shape of the cell, and anchor the nucleus and other organelles in place. Figure 1 shows how intermediate filaments create a supportive scaffolding inside the cell. The intermediate filaments are the most diverse group of cytoskeletal elements. Several types of fibrous proteins are found in the intermediate filaments. You are probably most familiar with keratin, the fibrous protein that strengthens your hair, nails, and the epidermis of the skin. Microtubules. As their name implies, microtubules are small hollow tubes. The walls of the microtubule are made of polymerized dimers of alpha-tubulin and beta-tubulin, two globular proteins. Figure 4. With a diameter of about 25 nanometers, microtubules are the widest components of the cytoskeleton. They help the cell resist compression, provide a track along which vesicles move through the cell, and pull replicated chromosomes to opposite ends of a dividing cell. Like microfilaments, microtubules can dissolve and reform quickly. Microtubules are also the structural elements of flagella, cilia, and centrioles. The latter are the two perpendicular bodies of the centrosome. In fact, in animal cells, the centrosome is the microtubule organizing center. In eukaryotic cells, flagella and cilia are quite different structurally from their counterparts in prokaryotes. As discussed below, flagella and cilia. To refresh your memory, flagella, singular equals flagellum, are long, hair-like structures that extend from the plasma membrane and are used to move an entire cell. For example, sperm, euglena. When present, the cell has just one flagellum or a few flagella. When cilia, singular equals cilium, are present, however, many of them extend along the entire surface of the plasma membrane. They are short, hair-like structures that are used to move entire cells, such as paramecia, or substances along the outer surface of the cell. For example, the cilia of cells lining the fallopian tubes that move the ovum toward the uterus, or cilia lining the cells of the respiratory tract that trap particulate matter and move it toward your nostrils. Despite their differences in length and number, flagella and cilia share a common structural arrangement of microtubules called a 9+2 array. This is an appropriate name because a single flagellum or cilium is made of a ring of 9 microtubule doublets, surrounding a single microtubule doublet in the center. Figure 5. You have now completed a broad survey of the components of prokaryotic and eukaryotic cells. For a summary of cellular components in prokaryotic and eukaryotic cells, see Section Summary. The cytoskeleton has 3 different types of protein elements. From narrowest to widest, they are the microfilaments, actin filaments, intermediate filaments, and microtubules. Microfilaments are often associated with myosin. They provide rigidity and shape to the cell and facilitate cellular movements. Intermediate filaments bear tension and anchor the nucleus and other organelles in place. Microtubules help the cell resist compression, serve as tracks for motor proteins that move vesicles through the cell, and pull replicated chromosomes to opposite ends of a dividing cell. They are also the structural element of centrioles, flagella, and cilia. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.